Okay, so the session is being recorded right now. Let's see, let's get started. Okay, so one of the main topics is muscle contraction, and you guys are gonna need to know the events that occur during the muscle contraction cycle, what happens at the neuromuscular junction, and all of that. So let's get started. Okay, so this thing right here is the neuromuscular junction. Muscular junction. And all it is just a neuron right here attached to a muscle fiber, which is down here. So this is what happens. Let's say you want to move your finger. Your brain sends that message down the axon. Okay, it creates an action potential. The signal down through here. Okay, now once the action potential reaches the axon terminal, this is what this thing is called. The end of the axon is the axon terminal. Okay, this activates calcium gated channels. Now, once these calcium gated channels are open because of the action potential, calcium rushes in. So calcium goes. And then the next step is this causes these vesicles to go here. They attach and they perform exocytosis and they release acetylcholine. ACH through the synaptic cleft. Okay. Now, once ACH is in the synaptic cleft, there are receptors on the motor end plate right here that acetylcholine attaches to. Now, once acetylcholine attaches to this motor end plate of the muscle, what happens is that sodium channels open now. Now, sodium channels open. This causes sodium to rush in, and now this creates another action potential that goes down the end of the muscle down into the T-tubule, right? This is right here is called the T-tubule. Down here into the sarcoplasmic reticulum. Now, once this action potential reaches here, the sarcoplasmic reticulum, there's something called the calcium. Again, it's another gated channel that releases calcium into the sarcoplasm. So here's the action potential. This opens up calcium channels. Now, calcium is able to be released into the sarcoplasm or the cytoplasm of the muscle cell, and this is what causes the muscle contraction. Okay, so just to review, the impulse travels down the axon terminal. Okay, voltage gated channels open, enter the terminal, allowing exocytosis of acetylcholine. Okay, into the synaptic cleft, like we mentioned, that little space. Now, acetylcholine binds to ligand-gated receptors on the motor end plate, and sodium diffuses into the muscle cell, creating an action potential that travels sarcolemma down into the T-tubule, where another voltage-gated calcium channel opens up, and calcium is released into the sarcoplasm, okay, from the sarcoplasmic reticulum. So the sarcoplasmic reticulum basically stores the calcium the most. Okay, and then from there, We're gonna get into the specifics a little bit later. But what happens is calcium binds to troponin. This causes a conformational change in the muscle cell. Tropomyosin moves out of the way, allowing the active site on actin, the small muscle fiber, to be exposed so myosin can attach to it and then cause the contraction. But we'll get into the cross bridge in a little bit. Any questions so far?
No, all good.
All right, cool. So this is muscle contraction. Perfect, Heather. Now, what happens during muscle contraction is, now, do we remember how we said there was acetylcholine that was released, right? There's a bunch of acetylcholine here. And then they are attached to these receptors. Now, how do they disattach from these receptors? Well, there's something called acetylcholine esterase. Terrible. Okay, acetylcholine esterase. And what that does, it just breaks down the acetylcholine that is in the synaptic cleft to get it out of there. Because if there's no more acetylcholine in the synaptic cleft, then it can't burst. Then the action potential cannot occur, so then you cannot continue contracting. So now you're relaxing. Okay, so acetylcholine esterase is one of the ways that one of the mechanisms of muscle contraction. Okay, now another way is, now remember how we said there was calcium in sarcoplasmic reticulum? That's what causes the contraction. Well, now calcium is taken out of the sarcoplasm and back into the sarcoplasmic reticulum. Questions? So for muscle contraction, those are the events. You have acetylcholine esterase breaking down acetylcholine. Okay, you have the calcium that is removed from the troponin. And then lastly, the last thing I didn't mention yet is ATP. Okay, ATP is added to the myosin head, and that causes the myosin head to relax. Okay, this is one of the only instances where you have ATP not doing work. It's just literally just causes the muscle to relax. Any questions? All right, moving on. How is the tropomyosin head exposed? Okay, so the tropomyosin head is exposed by calcium to troponin. This causes a conformational change, moving tropomyosin out of the way, allowing the myosin head to attach. Okay, next question is what is, you guys are gonna need to know the steps of this as well, but it's not too simple, it's not too difficult. Okay, so this blue thing right here is our actin filament. Right here is our myosin, the thick filament. So this is what happens. So now calcium has already been attached to troponin. You just don't see it in this picture. Okay, this causes the tropomyosin, this yellow thing. I mean, not yellow. What color is that? Like a beige longitudinal line right here. That is tropomyosin. Now it has been moved out of the way already. This is why this head is able to attach. So the myosin head attaches to actin. Okay? That is the first step of the cross-bridge cycle. Now you have ADP and inorganic phosphate. They are removed from the myosin head. This causes what we know as the power stroke.
Wait, Kevin, I'm not seeing the PowerPoint anymore. Am I the only one?
Yeah, let me know, guys, if she's the only one.
I'm still able to see it.
You're still able to see it? Let me see.
Okay, I could see it now. Reloaded it.
All right, cool. Okay, so the inorganic phosphate and the ADP are released from the myosin head, causing it to contract, or what is known as the. Okay, now the muscle will remain contracted unless ATP comes in. ATP is what causes the muscle to relax. So ATP goes back, it attaches to it, okay? Then ATP is hydrolyzed, which just means it's just broken down. Now this hydrolyzation process is what relaxes the myosin head, okay? ATP goes, it attaches here, ATP gets split apart, myosin head, talked about insufficient, waiting for calcium to do its thing, to attach to troponin, move tropomyosin out of the way so that it can go back and attach to the actin. And then this cycle continues for as long as there's a stimulus, as long as there's an action potential, as long as there's calcium in the sarcoplasm. And then it'll just back. ADP and inorganic phosphate are removed. This causes the power stroke. ATP is attached to the myosin head, so it can relax it. I mean, not yet. It attaches to the myosin head. Then it gets hydrolyzed. Then it relaxes, ready to be sprung into action for the. Okay, so this ATP right here is what we call the cocked position. Okay, just like a gun, you cock a gun so you can fire it. Same thing for this. The ATP cocks the myosin head so it can be ready to spring into action when needed. Does the cross bridge form? Again, calcium shifts tropomyosin out of the way by attaching to troponin. So calcium attaches troponin, moves tropomyosin out of the way. Next, define sarcomere, and that's all it is just the functional unit of a muscle cell or a muscle fiber. Okay, and all it is we'll get into detail, but remember each muscle fiber has functional units. There's something called Z lines. They're like little zigzags. So in between these Z lines, so from Z line to Z line is the sarcomere. Okay, that is the functional contractile unit. Okay, now he's asking you to draw out the general structure of a sarcomere. Now, you won't have to draw it out on the exam, but you may need to be able to identify the different regions of it. Okay, pretty simple. These are the two muscle cells right here. So the A band is the dark color. A, dark. And why is it dark? Because that's what is of the myosin thick filaments. Okay, next we have the I band, which is this light layer right here, and that is mostly composed of actin, or the thin filament. Another zone is the M line right here, and that is just the middle sarcomere. Easy, M line middle. Okay, and then you have the H zone, and it's basically the zone right here in between the M line where there's no overlap of the thick and thin filaments. So it's only myosin in this area. Okay, any questions? Those are the basic parts of it. Oh, and the Z disc right here. That one is pretty simple. It's just that any zigzag line you see, that is the Z disc, and that is the beginning, and that is the end of the sarcomere. Okay, so from Z disc to Z disc, that is your sarcomere. Okay, now they're asking you what is a skeletal muscle triad. Whenever they talk about a triad, just know it has to do with the neuromuscular junction, well, closer to the muscle cell actually. So remember that we said there was a T-tubule where the action potential crosses, goes down. Now on the two sides of the T-tubule, you have something called terminal cisternae. Okay, so these three things, 1, 2, 3, are what's known as the triad. And basically the triad is it receives the signal and releases the chemical ion, calcium. Okay, what is the name of the plasma membrane of the skeletal muscle? It's called the sarcolemma. Okay. Now, if they ask you what is the name of the cytoplasm for the skeletal muscle, that is called the sarcoplasm. Okay. So sarcolemma is the plasma membrane. Sarcoplasm is the cytoplasm. They just name it sarcoplasm for the muscle cells instead of cytoplasm. So that's where all the liquid and stuff is, and this is where the calcium gets diffused into. So what is a motor unit? A motor unit is one motor neuron innervating muscle fibers. It could be one, it could be 100, it doesn't matter. Okay, so neuron innervating muscle fibers. Any questions so far? A lot of these are repetitive because these are the main topics on the exam. It's the neuromuscular junction, the cross-bridge cycling, and all of that. So what is a neuromuscular junction? We talked about this plenty of times before. It's where a neuron meets a muscle cell. Okay. So what events occur at the neuromuscular junction? Well, we talked about it. Well, what we talked about is known as excitation, contraction. Okay, that thing that we talked about in the beginning that is known as excitation, contraction, coupling. So the excitation part comes from the action potential, that electrical signal that excites the muscle cells excites the neuronal cells, and then that action potential eventually reaches the muscle cell down to eventually cause the release of calcium and in turn cause the contraction of the muscle. So whenever you hear excitation, contraction, coupling, just know it's that whole process of the action potential reaching the axon terminal, releasing acetylcholine into the synaptic cleft, acetylcholine leading to the motor end plate of the neuron, creating another action potential that travels down the sarcolemma, the plasma membrane of the muscle cell, down into the T-tubule, releasing calcium into the sarcoplasm that then causes a contraction. So what is a motor neuron? A motor neuron is an afferent neuron that sends movement signals from the brain to the rest of the body or the periphery. You guys are familiar with afferent, efferent neurons, right?
Not gonna go over it.
What is a motor neuron? Yeah, of course, we'll go over it. Okay, let me make a correction right here.
Because he kind of went over it today, but he like runs through stuff.
Efferent. Sorry. So make that correction, guys. A motor neuron is not an afferent neuron. It is an efferent neuron. And I'll explain what that is. Okay, so an afferent neuron. So let's say you walk out into the cold. You feel cold. Why do you feel cold? Well, you have sensory receptors on your skin that detect change in temperature. Okay, so from these sensory receptors, they can be anywhere on your body. This is one example. So from the receptors of your body to your brain, we call that afferent. So whenever you hear afferent neuron or sensory neuron, then they're the same exact thing. Okay. Now, what do? How do you respond to this change in temperature? Well, now then your brain sends a signal from itself, from the brain, back out to whatever structure that is associated with keeping your body temperature level. Okay, now this signal from the brain back to the rest of the body is called an efferent neuron. And this is also called a motor neuron. So that's just it when they mean afferent neuron, efferent neuron. Afferent neurons send information from the rest of the body to your brain. There, when your brain decides to take action, your brain sends a signal from itself to the rest of the body, and that is known as an efferent neuron. Another way you can remember it is efferent is exiting. They both have E's, exiting the brain, efferent. Afferent, you just got to memorize that. If you know it, then you'll know afferent. Okay, so afferent is sending signals to the brain, efferent is sending signals away from the brain. And afferent neurons are sensory neurons, efferent neurons are motor neurons. Questions?
Good so far.
All right. What is excitation, contraction? We talked about that. You guys want to go over it again?
No, that's fine.
Yeah, that's fine. You can continue.
All right, thank you. Just let me know if you guys need me to go over anything, okay? If you're confused, stop me and I'll gladly explain it to you. Okay, next, define wave summation. It just, so this is talking about the kind of signals your muscles receive when you're about to contract. And wave summation is just a muscle contraction that happens consecutively without the muscle rest. So it looks like this. So let's say the muscle has a contraction and it goes back to rest, right? So now on the other hand of it, let's say your muscle contracts, it's about to relax, but then there's another signal that comes, oh shoot, I can't relax. It goes back up, it contracts again. It goes back down, it tries to relax, but it comes in, it can't relax, it goes back up. So as long as there's a signal coming, the muscle will not relax and it'll just keep on going up and down. And then eventually it'll relax once the signal stops. This is a wave summation. I have a prettier picture, but it's on the next page. They define muscle fiber. When you hear muscle fiber, that is the muscle cell, okay? Myofibril are the muscles, are the filaments found in muscle cells. So your actin and your myosin. Those are the myofibril. When they talk about myofibril, that is actin and myosin. And remember, actin is the thin one, myosin is the thick one. Okay, so compare and contrast a sarcomere, a muscle fiber, and a myofibril. Well, to compare them, all of them work together to cause a muscle contraction, okay? So the myofiber is part of the muscle fiber, and then sarcomeres, a bunch of sarcomeres also make up the muscle fiber. So how can I explain this? So you have your little actin and myosin fibers, actin and myosin fibers. Okay, these are called the myofibrils. Now, a bunch of these together make up the muscle fiber or the whole of the muscle cell. And then inside the muscle cell, you have functional units called sarcomeres. That's basically. What are the two types of neurotransmitters found at the neuromuscular terminal? The main neurotransmitter is acetylcholine. Okay, quick question for you guys. How is acetylcholine released into the synaptic cleft? Causes that response? Anybody? Okay, so remember that acetylcholine is released through exocytosis. Why? Because you have an action potential that arrives at the axon terminal. Okay, and then what happens next is activates calcium voltage gated. Calcium rushes in to the axon terminal, and then because of this influx of calcium, acetylcholine is released into the. Okay, make sure you guys know those steps because that's the huge part of the exam, okay? Okay, we already did these. Just the events of the neuromuscular junction. Well, the neuromuscular junction is itself, excitation contraction is one of the processes that happens in the neuromuscular junction that eventually leads to the cross bridge or the actual contraction. Okay. Now define twitch. Twitch is just a muscle contraction during period. There's just one stimulus. Okay, now as I said, we have wave summation where the muscle does not go into its resting phase, right? Just there's this constant input of stimulus, and the muscle is never able to relax. Okay, and then here you have something called incomplete tetanus, and then complete tetanus. In complete tetanus, you have a full influx of stimulus one after the other. Things are just stimuli, just one after the other, and this causes a full contraction. It recruits every single muscle. Now, for incomplete tetanus, you have the stimulus at different time intervals. This doesn't really cause a full, complete contraction. Okay, now we're gonna get into isometric contractions. So iso means same. So the muscle is producing tension, but it's not changing its length. So if we look at this graph right here, okay, the blue line is the tension, and then on the bottom, that is the length of the muscle. So here we're inducing tension, but the muscle is not going anywhere. It's not moving, but that's called isometric contraction. So let's say if you try to go push up against the wall, like you're trying to push the wall, you can try as hard as you want. Your muscles are not moving, but you're still creating tension and doing work. Isometric. Now for isotonic contraction, you have tension is produced when the muscle contracts or it relaxes. So you have two types of isotonic contraction. You have eccentric and concentric. Eccentric is just when the muscle elongates or stretches, and then concentric is when the muscle shortens. So let's say you're doing a bicep curl. You bring the weight closer to your hand, to your head, to your body. That is a concentric contraction because the muscle is shortening. Now, when you finish that rep, you gotta lower the weight back down. The muscle is elongating, and that is known as eccentric contraction. Define concentric contraction. We just did that. So concentric contraction is when the muscle is shortening. Okay. To compare and contrast, we'll go over this again. So eccentric contraction, muscle lengthens, eccentric elongate. That's a way you can try to remember it if you're having trouble with it. Concentric, you're concentrating, you're focused, muscles are tight, they're short. Okay. Isometric contractions, muscle remains the length. There's no muscle movement, but you still have tension being produced. So isometric contractions usually occur when the load or the weight is greater than what you can exert. So a wall, for example, you won't be able to move a wall, but your still muscles are producing tension, trying to move that wall. Not gonna go anywhere. Okay, now you have fast twitch muscle fibers. He doesn't, oh, and slow twitch, yes, he does talk about it, never mind. So two muscle fiber types. Fast twitch muscle fibers, it's really in the name. Fast release of energy. Okay, you're sprinting, you need those muscles to work really fast, to release energy fast just so you can do that work. fast twitch muscle fibers take over. Okay, and this is in anaerobic conditions. No oxygen. Slow on the other hand, is for long-term release of energy. So let's say you're doing a marathon, you're jogging for 5 miles, whatever it is, any long-term work, you have your slow twitch muscle fibers providing that energy for you, okay, and doing that work for you. Twitch fibers take over in an oxygen-rich environment or aerobic condition. Okay, now here's some different cycles in the body that allow us to produce energy. Okay, you won't really have to know the details of them, just the general idea of it. Okay, so the Cori cycle is one of them. The first 15 seconds. Okay, you're doing work, your body quickly needs to produce energy. So what it does is it takes what is it? Is CP, creatine phosphate. Okay, takes creatine phosphate along with ADP. What comes out is ATP, creatine. Nah, guys, I am so sorry. I'm talking about the creatine phosphate cycle. Why didn't you guys stop me? Let me restart that. So what I was just talking about is the creatine phosphate cycle. We'll go back into that later. The Cori cycle, I'm sorry, all it does is converts lactate back into glucose. And this is very important because you guys lift weights and then your muscle really starts to hurt, it starts to burn. That is lactate buildup. Now that is for the body. Your body needs to find a way to convert it back into something safe. This is where the Cori cycle comes in. It takes the lactate or the lactic acid and it converts it back into the glucose. Okay. Now you again have the lactic acid. So again, this is anaerobic conditions when there is no oxygen. So again, you're lifting weights, you're doing it very fast, you're not able to input as much oxygen. What is your body gonna do? Is it gonna just stop working? No, it kicks in the lactic acid. Glucose is converted into lactic acid to create ATP, energy. So all of these cycles create energy, okay? So lactic acid creates energy when there's no oxygen, creates lactic acid, and then the Cori cycle takes back this lactic acid and turns it into glucose. Next, we have anaerobic fermentation. Okay, so anaerobic, no oxygen, and fermentation. That part. So a lactic acid cycle is a type of anaerobic fermentation. Okay, whenever there is no oxygen present, we have anaerobic fermentation occurring. So if there is no oxygen, fermentation occurs. This is usually in plants, but we have it as well. Plants produce alcohol as a result of fermentation, and animals, we produce lactic acid. Okay? Now, the creatine phosphate cycle, talking about in the beginning. This is what happens in the first 15 seconds. Okay? We have creatine phosphate is used to create ATP. So these two things come together, okay? The phosphate and the creatine phosphate attaches to the ADP to create ATP, and then now the creatine phosphate simply just becomes creatine because it just donated its phosphate group to the ADP. Any questions? So the creatine phosphate cycle is the first process that occurs when there is no oxygen, okay? If there continues to be no oxygen, creatine phosphate cannot continue to work. So what kicks in? The lactic acid cycle kicks in after the creatine phosphate cycle. And this is, again, if there is no oxygen. So if you're lifting weights, something very, very intense. Okay, but on the other side of it is if you do not doing too much intense work, we have aerobic. Aerobic respiration. Okay, but just make sure you guys focus on these, the Cori cycle, because you might ask questions about it, but very basic. Okay, so Cori cycle, it just converts lactate back into glucose, lactic acid. When there is no oxygen in the environment, your body produces lactic acid to continue giving you energy. Again, this is long-term energy, is very, very painful. Next, we have anaerobic fermentation. Again, the presence of no oxygen. We talked about the lactic acid is a the lactic acid is a type of anaerobic fermentation. And then lastly, we have the creatine phosphate cycle, which in the first 15 seconds of the work. Okay, now let's get into the types of muscles. Let's see. What is the function of skeletal muscle in the body and where is it generally found? Well, the function of skeletal muscle is to provide heat, support, guards entrances and exits, and protects your internal organs and provides as well. There's an important note to make about the entrances and exits. Okay, the muscles that guard entrances are usually certain. Okay, the muscles that guard entrances are circular. The muscles that guard entrances are circular. And let's not repeating this a bunch of times. One more time. The muscles that guard entrances are circular. Okay, remember that. Next, we have cardiac muscle. Where is it found? Strictly found in the heart. Okay, and all it does is functions to keep the heart pumping. Now, last type of muscle is smooth muscle. Okay, the function of it is just to move blood around, move food around, for peristalsis or contraction. So when you eat something, how does it go down your intestines? Well, peristalsis is just that's what it's called, the contraction um, of the large intestine, the contraction of the small intestine. All that is peristalsis. That movement is called peristalsis, and they're found in the intestinal linings, capillaries, veins, arteries. Some things to note about smooth muscle: they call it smooth for a reason. It does not have striations. like the cardiac muscle and the skeletal muscle. Okay. Now we're gonna get into muscle types. Epimysium is just the top layer of the connective tissue. Epimysium covers the entire skeletal muscle. Topmost layer. Now, inside the muscle cell, you have something called fascicles, which are just bundles. My screen is freezing. Can you guys hear me?
Yeah.
All right, cool. Sorry, my
Yes.
computer just froze for a sec. Okay, so we talked about the epimysium, which is the connective tissue that surrounds the entire skull. Then we have the perimysium, which just surrounds the muscle fascicles, which are just bundles, okay, of myofibrils. And then each individual muscle fiber, the muscle cell, bundles of muscle cells. Now, each individual cell is surrounded by something called the endomysium. Okay? A synergist. A synergist is just a small muscle that supports the movement of the muscle in the work, or what we call the agonist. Okay? So if you're, let's say, this is might not be a real example, but let's say you're trying to contract your bicep, right? You're trying to lift some weights. Now you have some secondary muscles. I'm not too sure of their names, but they assist the bicep in lifting that weight. Okay? Now, antagonist, it's the muscle that opposes the agonist movement. So the agonist is the muscle that you're trying to work. Okay? The muscle doing movement. The antagonist is trying to oppose that movement. Okay, pretty simple. So for example, again, back to the bicep curl, you're curling the bicep. So your bicep is the agonist, right? The antagonist, the muscle opposite that can potentially oppose it if it chooses to, is your tricep. So while your bicep is contracting, your tricep is relaxing. While your bicep is relaxing, your tricep is contracting. Okay, the fine agonist I just did. It's again the muscle that produces a particular movement, and the antagonist is what opposes that movement. What hormones that increase muscle mass? Growth hormone, epinephrine, testosterone, and thyroid hormone. Okay. Okay, so what are the primary muscles used for facial expression? you have your mentalis, your orbicularis oris, which is the muscle around your lips, your risorius. Note about the risorius is the smiling muscle. Okay, guys, please take note of this. The risorius is the smiling muscle. Okay, it's just a small little muscle. Let's say this is your lip. This is bad lips, whatever. You have a little two muscles right here, tiny projections that are on the outside of your lip, the corner of your lip. I think I have a better picture, but anyways, those two little muscles are what's known as the risorius. Okay, you have your zygomaticus major, zygomaticus minor, your buccinators, which is part of mastication, and then your depressor anguli oris. Okay, so what are the extrinsic eye muscles? Pretty simple. Four rectus, two obliques. Okay, if you guys can remember this, the is easy. Why? Because for the rectus, all you have is up, down, left, right, superior, inferior, lateral, medial, and then for your obliques, you just have up and down, superior, inferior. Now, for the rectus muscles, it's important to know how they move the eye. Okay? Now, the rectus muscles are pretty simple to remember. Like they move the eye in the direction of the name. Superior rectus moves your eye superior. Inferior rectus moves your eye down. Lateral rectus moves your eyes laterally or away from the midline. I don't want to say right and left because it's right for the right eye and left for the left eye, lateral movement. The midline of the body. And then the medial rectus moves your eyes towards the middle part of your body. So let's say you're looking in. Okay, so rectus moves the eye in the same direction as the name indicates. Now your obliques that are a little different. They move the eye in the opposite. So your superior oblique moves your eyes down and lateral, and then your inferior oblique moves your eyes up and lateral. Okay, so rectus, same direction. Oblique, opposite direction of the name. Okay. So these are your extrinsic eye muscles. So your rectus, so this is your superior rectus up top, moves your eyes up. Your inferior rectus is down here on the bottom. Okay. And then you have your lateral rectus side, and then your medial rectus is right here behind it. Okay. And then here are your obliques up top and on the bottom. So again, the inferior oblique rotates your eyes up, opposite to its name. Superior oblique rotates your eyes and laterally. Okay. Okay. Now, what is the masseter and where it is located and what's its function? So the masseter is one of the muscles used for mastication and is near the mouth. Let me see if I have a picture here. No, sorry, shoot, no picture. But anyways, you guys could look that up. It's this big muscle. Okay, big muscle. That is where the orbicularis, and I'm sorry, that is where the masseter is. Okay. Now list all the circular muscles located in the face. So recap, what do circular muscles do? What do they guard? What do circular muscles guard again?
Your eye.
Entrances. They guard entrances. Okay.
You're right. You said it like 3 times.
So
Guard and
yeah, exactly. You have two circular muscles in the face. Orbicularis oris, which is your mouth, and then orbicularis oculi, which are your eyes. Okay, two circular muscles on your face. What is the quadratus lumborum? Where is it located? Again, I didn't really include these pictures because he doesn't really ask too much about them. They're not really labeling on this exam. It's just knowing what they do and their function. So the quadratus is located on the pelvis. It attaches to your lower ribs, and all it does, it keeps your body upright. Okay, it's one of the erector spinae muscles. So whenever you need to stand upright, when you need to bend over, that is your quadratus lumborum. Okay, it holds up your whole torso. Where is your iliocostalis? Where is it located? It's another erector spinae muscles, and that's just for support and posture. Whenever you see here erector spinae muscles, it just keeps the spine upright and erect, basically. Okay, and they call it iliocostalis is because it attaches on the ilium of the pelvis, and it goes to the costa, aka your ribs. So it's up and down the length of your whole entire spinal cord. Okay. What is the longissimus dorsi? Again, another one of the erector spinae muscles, so not to be confused with latissimus dorsi. Again, located near the spine and supports the posture. So compare and contrast these three muscles we just talked about. There's really no contrast except for their location, but their function basically is the same. Keeps the muscle, it keeps your upper body upright and provides support. Next is your sternocleidomastoid. That is located in your neck, and does it just rotates your neck left and right. Another muscle of the neck is known as the platysma, and that's like a sheet. Okay, and all it does is just serves to lower your lower jaw or your mandible, and it tenses the muscles of your neck. So of the platysma. What is the genioglossus and where is it located? So this is part of your tongue. Okay, you have three parts of your tongue. And the main part, the one that your professor focuses to choose on, is the genioglossus. Then you have the hyoglossus and the styloglossus, but those are not as important for this exam. Okay, and all it is the majority of the structure of your tongue. So when you stick your tongue out, what you're seeing is the genioglossus. Okay. Now, what are the muscles of the rotator cuff and of each muscle? 4 muscles associated with the rotator cuff. your supraspinatus, your infraspinatus, your teres minor, and your subscapularis. So your supraspinatus works to abduct your arm, which means take it away from your body. Infraspinatus is for external rotation, as well as teres minor, again for external rotation. And then lastly, your subscapularis, which is used for internal of the shoulder. Okay? So here we are. So this is the posterior side. Or the back side. And then this right here is the anterior side, or the front side. So your subscapularis is the one that is on the front. Okay, it covers the majority of the front of the scapula. Remember, part right here is called the subscapular fossa. That is where the subscapularis muscle attaches. And then your supraspinatus, again, this is the of this is super exactly. And then below it, you have your infraspinatus. Okay, teres major, teres minor is right here below the infraspinatus. What are the structures that increase the stability of the hip joint? We have iliofemoral, our pubofemoral, and our ischiofemoral ligaments. Okay, what happens to the muscle during rigor mortis? So rigor mortis is after you die, your body becomes extremely stiff. And we're going to explain why this happens. So when you die, your sarcoplasmic reticulum breaks down. That releases calcium into your muscle cells, sarcoplasm. This causes your muscle to contract. Why? Because calcium attaches to troponin. Tropomyosin moves out of the way. Remember that how we said the myosin head was already locked and loaded? So boom, calcium moves troponin, moves tropomyosin out of the way. Myosin it attaches and it contracts the muscle. But you're dead. There's no ATP. You're not producing ATP. And remember how we said ATP is what relaxes the myosin head because it's split apart? Well, now that you can't do that. Your body is not producing ATP, so your muscles stay contracted and stay stiff. So that's what happens during rigor mortis. Pretty cool stuff. All right, now these last miscellaneous questions. We talked about muscles of the rotator cuff. We're good with that. Muscles involved in chewing and the mandible muscles of the leg. Okay. So these are some miscellaneous stuff at the end. As I took a look at your exam. And these are some of the questions that were missing. Okay. What are the muscles of the leg? Let's start with that. You have four muscles. Your rectus femoris, which is this big muscle right here. Beneath the rectus femoris, you have the vastus intermedius. Okay. Now, the famous teardrop muscle, the one that everybody wants, myself included, vastus medialis. Okay. Now, on the outside of the leg, you have your vastus lateralis. Okay, these names are pretty easy. Okay, let's go to the last page, and we're done, and I'll open up question for questions. Okay, so what are the muscles of your pelvic floor? Some important ones. Your pubococcygeus, your iliococcygeus, okay? This is bad right here. This right here is your iliasis. And those are the main muscles of the pelvic floor. Coccygeus, iliococcygeus, which make up something called the levator ani. Both of those together make up the levator ani. Okay. And that is it. You guys have any questions? I'm going to start recording.